I am now going to pass it over to Sue Weintraub from the University of Texas Health Science Center in San Antonio. And she is also going to talk about DIA. So I'm excited to pass it over to her. So let me do that. And Sue, I will pass it to you.
I'm getting there, just finding the right. Okay. Thank you very much, Jacob. I'm delighted to be here and presenting to this group. As you can see from the title of my slide, I think DIA is absolutely astounding. And I consider it the most exciting technology I've used at least the past 20 years. We've been doing DIA analysis for at least 2 years, 2.5 maybe. Really waited until the Scaffold product was available because I wanted a clean and easy workflow that could be used in the context of a core laboratory. I'm showing here our general process method. This experiment was now done a few years ago, closer to the beginning when we optimized our scan settings. It was 4 experimental groups and 3 replicates, just showing using S-traps for sample prep, the MS analysis of 2-hour gradients, the Scaffold DIA processing. Now, it's a day, but we do, as the samples are acquired, convert them as needed to the .dia format. But the total time was only 5.25 days, and we got 6,000 proteins. Of note, here we were using 6 gas phase fractions to make the chromatogram library. We're now using 3. Fairly early in our use of, or really exploration of DIA, we had this project come in from Alex Bishop, where they wanted to look at differences in the chromatin fraction of cancer cells after the cells had been manipulated in a certain way. We have traditionally done most of our mass spectrometry and proteomics using gel-based, what we would call gel CMS. And it really is very nice to be able to look at a sample on a gel to get a sense of the complexity and relative quantities. In this case, we didn't have a protein concentration at this point, but loaded different volumes and could see quite a few proteins and fairly evenly distributed. Then using our standard process, gel is only run about 1.5 centimeters, cut the gel lane into 6 slices. Each slice gets reduced and alkylated, digested with trypsin, analyzed by MS this was for DDA analysis. Then we use Mascot for database searching, and then assembly into Scaffold. These were run on Scaffold 4. Now the new version is Scaffold 5. And you see the layout that I'm sure you're familiar with. And I'm just calling it cell 1 and cell 2 because this work isn't published yet. Showing in regular Scaffold 4 samples view the total spectrum counts. You can do a t-test as shown here. And the green is representing statistically significant with multiple testing correction. You can notice in the pink area, there were 1,766 proteins identified. If I sort by smallest to largest p-value, you can clearly see that there's some striking differences between the experimental groups. Scrolling all the way down, there were 788 proteins that were significant with equal or less than P022. That's a fairly large proportion. So we really had already given the investigators some interesting information, but decided this would be a perfect test for DIA. And here is the samples view in Scaffold DIA. And what we always look at first is the run order. Our samples are blocked and randomized to minimize any kind of systematic error. And it's always very comforting to look at the run order and not see a pattern, for example, a constant decrease in quantity or increase or something that would indicate an instrument or processing problem. The next thing we usually do is the principal component analysis, because I can't wait to see, are there going to be some interesting differences? And in this case, not surprising that there's very striking separation between the two cell types. And there's even two dots right on top of each other here. Now, you'll notice that I've highlighted exclusive intensity, and this is an absolutely key point. It's also fairly important looking at non-shared peptides in spectral counting, but here it's really critical. In the similarities view, or similar proteins, which I really love having in DIA, and you see that also in the LFQ, what you notice for PLRG1 is there's quite a few peptides that were identified and quantified. Nothing is shared, so you don't have to worry about this protein. But for PPB, and that's a serine 3 protein phosphatase, there's quite a bit that is shared. And what you see is there are three forms. These are the catalytic subunits of B, A, and G. And A has the most that is exclusive to this protein. As you will see, some places call this unique, but really exclusive is the most appropriate name because it's exclusively assigned to this protein, while B and G have less proportionally. Well, why does this matter? Look at the exclusive intensities. These are the summed intensities for all of the peptides assigned to the protein. And you see that there's a very large difference between the quantity of A and B and G, and then a different proportion in the two cells. If you counted all of these peptides, say for isoform G, you would get a noted overrepresentation or overemphasis. The quantity would not be correct because you have something that's much more abundant that's being counted. So it's really important to use for all of the quantitative data only the exclusive peptides. Now, also something that I love, it's been an important feature of all of the Scaffold products, is I like to look at data, and I'm not happy just getting a report of an Excel spreadsheet where I can't examine what's happening in the spectra. In DIA, you're not going to have a standard spectra that you're used to from DDA, but what you can see is the peptide fragments that are coeluding. And these are the ones that are being used for quantitative analysis. The ones that are colored are the ones that are counted, but the others you can see are also coeluding. The precursor is shown here, but it's not used for quantitative analysis. But what this is important to think of is, picture a DDA spectrum where you have a pattern, and you expect that that pattern is going to be the same for other samples that have that same protein and that same peptide. The proportion of those peaks should be the same. And that's what you can see in the fragment intensities view, where even though the absolute quantity of each of these is different, the relative proportion is the same. And this gives us confidence in the quantification and identification. Now, you don't do this, obviously, for every one of thousands of proteins, but when you have a protein that's of particular interest, and especially if it's low abundance, you want to be able to examine it yourself and be sure you have confidence in it. All right, now back to the results. Again, going to do a t-test and sort by p-value in the same way. Green here is significant with correction for Benjamini-Hochberg multiple testing. And you really can see the notable difference in the relative quantities of this group. I'm going to scroll down again like I did for the spectral counts. Now there's 2,465 that are significant. Notice here, the ones in gold, those are less than P05, but do not pass the cutoff for Benjamini-Hochberg multiple testing correction. Then I wanted to see, here's it showing in the log2 screen so that you can really get a clear indication of what major difference there are in these two cells. But I wanted to see what was the comparison with the DDA data. And this is the set that I had just shown you. Going to highlight two specific proteins, DYHC1 and TCP4. And shown here is the corresponding DIA data. These are the same lysates. Obviously, they're different samples because the DDA was from a gel CMS and the DIA was from a single S-trap digestion and sample prep. And what's really notable here is we had two spectral counts in each of these cell twos. You do see a good, reproducible, and fairly similar intensities. But here we had 0. And does that mean really nothing? No, it's just a lower relative abundance, and it did not trigger any fragmentation spectra. Likewise for TCP4, here we have, you have a higher number of spectral counts, and the absolute abundance is higher, not absolute abundance, but the summed intensities from the DIA peaks. But over here you had 2 and then none. But in fact, it just meant that fragmentation wasn't triggered here. And if I needed any convincing of how great DIA was, this sample set made it very clear and to the investigator. And there it is in the log2 form where we're looking at the relative to cell 1. Now this was a very interesting set that we ran by DIA. And here I'm not showing you the run order, but it was a set that was from male and female mouse neural stem cells. And the experimental design was there were 3 mother mice, and then from each mother, there was a male and a female that was selected. The neurostem cells were acquired and put into 3 separate batches for each one. And then it received either vehicle, dose 1, or dose 2 across the 3 sets. This is the PCA for that group. And you'll notice a really amazing clustering because it turns out that it clusters by mother and by sex of the mouse. And what this shows you is that if you hadn't looked at them separately and you just pooled together by sex, by all the mothers as a separate experimental group, you're not going to see any statistical differences. But instead, if you do a multi-level statistical test within a mother and within sex, then you get to start seeing what the true differences are relative to the doses of the drug. So this is showing an amazing aspect of being able to do the PCA and associate the changes with the different groups. Now, in this set was some prostate cancer cells that the investigator had done some knockdowns on. And first, again, I'm looking at the run order. And in the log2 fold change view, you can really see the patterns well. And there doesn't seem to be any problem related to run order at all. Now into the experimental groups. This is the wild type. Nothing is done, no treatment. This was a sham treatment. This group is a knockdown of a particular protein, one set and another. And it was pretty disturbing because it didn't seem to fit into the experimental groups the way the investigator thought. Doing a PCA, what we could see is they really didn't seem to fall well. Now, this is the completely untreated, and this group is the sham, so that's not too bad. But these others really don't behave the way we thought they should. And seeing here, this was set 1. So this is designated 51, 1, and 2. And then here's the 49th. And I said to the investigator, have you done Western blots on this? How did you verify about your knockdown or knockout? And in fact, here they are. And 59, 1, and 2 were really knocked out, but 59, 3 wasn't, and the other 49s. And so I'm not sure what was happening at this point. And then she started thinking, she says, oh, well, each one was a separate clone. And it surprised me that she hadn't done a Western blot on each one first before submitting the samples, but she's gotten some very interesting information on differential levels of knockdown. But the mass spectrometer doesn't lie, so we could tell that something wasn't the way she expected. And here's some fairly recent data that's very exciting to me. This is in a urothelial carcinoma cells, bladder cancer, where the cells are isolated from individual tumors from separate subjects. Again, looking at the run order, making sure that there's no problem. Looks fine to me. There were 4,816 targets. Now these were cells from, again, each one is a separate subject. The investigator thought that there was going to be a difference based on the grade of the tumor, but there was no apparent correlation between tumor grade and relative protein quantity. When we looked at the PCA, you can see kind of interesting pattern. So I arbitrarily picked L, which is left for this group, the one that's in the middle here, and the ones on the right, just to see what would happen. And if you do a t-test, and this is comparing L versus R, what you see is a large number of statistically significant differences with Benjamini-Hochberg multiple testing correction. And this was quite amazing. So immediately, and I will say that Brian Searle directed me to not jump to conclusions that these were biologically relevant. So we went back and had the investigator look at all the different aspects of sample acquisition, the tumors, how they were prepared. And really there were no differences that they could find that were in any way related to sample prep. If you take this data set, then you can very easily export into Excel. And excuse me, I jumped ahead, but I wanted to show you this volcano plot of the data, which strongly shows a large number of significant differences. And if we pick this spot and look at the results, you see it's very convincing that there's something quite different about this protein and many others in the R compared to the L. Exported the results into Excel and then divided into the proteins that exhibited a 1.5-fold higher level of change versus up versus down, and submitted this to Reactome analysis. Reactome, if you haven't used it, is a fabulous site out of the UK where you have no charge and you can do pathway analysis, and it's especially powerful for human and mouse and rat also, but especially for human results. This is the foam, Reactome image, where the size of a block corresponds to the number of reactions of proteins inside. And the lighter the yellow, the more of your proteins that you submitted that are overrepresented. And we can see here that there are some very clear pathways that are overrepresented by these tumor cells. That's the ones that are 1.5-fold down, and in 1.5-fold up, there's different reactions. I've often found that this is, there's a difference between up and down that's worth looking at them separately. Now, this is something that we do for all the investigators who submit samples. I'm happy to work with them and do the interpretation because many have no experience in proteomics at all. And it's part of what I feel is essential from a core to be a collaborator and not make it as a service. Now, this investigator is intrigued by the results. We're planning now to analyze a very large number of samples so that there can be enough population in the different characteristics so that we can find statistical significance or biological relevance that related to an attribute we can understand. I have to thank Sam Pardo and Dana Moeller. They have done always a great job, but especially during the pandemic, they have been working split shift and keeping it productive, and they're just extraordinary in their work. I will also thank the Health Science Center and the Mays Cancer Center, NIH and University of Texas System. I perhaps didn't mention again, those were all run on Illumos, and the UT system paid for that Lumos and Proteome software, and it's been wonderful working with them. So thank you, and I'll be happy to answer any questions now, or anyone who would like to look at data with me in the future, we can set up a Zoom session and I'll be glad to show you and or look at yours. Thanks.
Excellent, Sue. Thank you very much for
I realized I didn't have my video on, but that's okay.
Thank you very much for giving that presentation, both on looking at some interesting data and using Scaffold DIA to analyze that data, and using Scaffold DIA to kind of figure out that something was wrong, and then reaching back out to the investigator and having them do a little bit more digging. And it's cool that not only were you able to use the program to get some results, but you were able to use it to do some kind of troubleshooting there. So we like to see stuff like that.
Right. I can tell you there's a data set running right now remotely on the lab, and I hope it'll be finished by the time we're finished and I can get to see some even new data.
Awesome. Cool.
Yeah.
Cool. All right.